Okay, good afternoon, everybody. We have got over 130 attendees at the moment, so let's kick off. So this afternoon, we'll be hearing some very interesting presentations from Dr. Claire Pallis and from Drs. Ian Wilson and Dr. Eric Olinger. This is the April Research Seminar Series being run by Genomics England. And just to let everyone know, this will be a recorded seminar, so it will be available afterwards for everybody. We record all our seminars, so they're available to view afterwards. And so the first talk will be from Dr. Claire Pallis. It will be on the germline loss of function variants in the base-excision repair gene, MBD4, caused Mendelian recessive syndrome of adenomatous colorectal polyposis and acute myeloid leukemia. And so after this talk, we will have a chance for Q&A. And then we will hear from Dr. Ian Wilson and Dr. Eric Olinger, and they will be presenting on exploring the molecular genetics of end-stage kidney disease within the 100,000 Genomes Project. And so both these talks that we will be hearing today have been made possible by the 100,000 Genomes Project data. And so this data is available and accessible by anybody at an academic or healthcare institution. And you can apply to join on our website, and there'll be a link available in the chat function of the Zoom. Also, we very much welcome research in industry, and they can join via the Discovery Forum. Again, there'll be information via the link. And so we hold these research seminars on the last Tuesday of every month, usually from 2 p.m. to 3:30 p.m. So our next one will be on the 25th of May, and we'll be welcoming speakers Dr. Yiting Zhang from Velo Health, as well as Dr. Matt Murray and Dr. Patrick Tarpey. And so you can find out more about our research via our Twitter on at GSIP team, that's the handle. And you can find the seminars that we have via the seminar videos link. And you can also hear more on our research and the work we do via our monthly podcast called The G Word, and that's hosted by our fantastic CEO, Chris Wigley, often with some very interesting guests. And also our research projects can be viewed via our research portal. And so everything I've just mentioned, you can view it via, I believe, some links that Sam is posting in the chat. Great, we are almost at 200 attendees. So I say let's kick off with Dr. Claire Pallis's talk. So Dr. Claire Pallis is currently leading the gastrointestinal cancer genetics group at the Institute of Cancer and Genomic Sciences under University of Birmingham. Her group currently investigates predisposition to esophageal adenocarcinoma and colorectal cancer. And they are also looking within those under the age of 50 and their tumors, and comparing them to patients diagnosed at older ages. And so I'm very excited to hear from you, Dr. Claire Pallis.
Thank you very much for that introduction, and thank you for the invitation to talk to you all about our work where we've been identifying germline loss of function variants in an enzyme involved in base-excision repair, MBD4. And we've identified a rare Mendelian recessive syndrome characterized by adenomatous colorectal polyps, and acute myeloid leukemia. And I'd like to begin my talk with the acknowledgement slides, as this has really been a big team effort, as is so often the case with rare mutations and rare causes of disease. So in particular, I'd like to acknowledge the work of people in my group in Birmingham, so Sarah Galavotti and Helen Curley and Laura Jedwin. We've also worked very closely with Rashada Dever's group in Nijmegen, and in particular with her PhD student, Judith Grolman, who's done a lot of the work that I'm going to show on the heterozygote carriers and their tumours and the mutation signatures seen there. We've also worked closely with Ian Tomlinson, who's now in Edinburgh, and he's the CI of the colorectal tumour gene identification study, also known as CORGI, where the patient that I'm going to describe at the beginning of the talk was first identified. A group in Melbourne, led by Ian Majewski, published a paper a couple of years ago now that identified patients with AML with biallelic loss of function MBD4 mutations. And we've worked with this group who've now, since their publication, gained access to colorectal tumour samples from these same patients, and have done a lot of work to characterise those that I'll describe as well in the talk. And then others listed here have provided access to samples as we've tried to work out how common mutations in MBD4 are, both monoallelic and biallelic. And in particular, the 100,000 genomes data has been really important for that part of the work in establishing how common these mutations are, and also what their role might be in sporadic cancers too. And the work actually also began in a project that preceded the 100,000 genomes work in terms of bringing whole genome sequencing to the NHS. And so I'd like to acknowledge Jenny Taylor and the members of the WGS 500 team. Also on this slide are members of the colorectal GSIP who've worked hard in analysing all of the data that's been generated by 100,000 Genomes Project for colorectal cancer patients as well. So our work began in trying to understand some of the novel or new genes that might explain predisposition to colorectal cancer and colorectal polyposis. So as this slide shows, nearly 3/4 of colorectal cancer cases are thought to be sporadic, and a small number of colorectal cancer cases can be explained by high penetrant mutations in genes such as the mismatched repair genes, MLH1, MSH2, that lead to Lynch syndrome, and mutations in the APC gene that lead to adenomatous polyposis coli. There are also rarer syndromes characterized by recessive mutations in MUT2IH and NTHL1, which leads to polyposis and multi-tumor phenotypes, particularly in the case of NTHL1, and dominant conditions caused by mutations, single mutations in the polymerase epsilon and polymerase delta encoding genes. So as I mentioned, this project began prior to the 100,000 Genomes Project, where we submitted 35 probands, all with multiple colorectal adenomas, to a project called WGS 500 that was run at the Wellcome Trust Centre for Human Genetics in Oxford. And having obtained the whole genome sequencing data, we prioritised coding germline variants with a high probability of a loss of function effect. And whilst looking for recessive mutations, we identified the mutation I'm going to describe today. So this was a search looking for homozygous or compound heterozygous mutations in genes where there was a high probability of a loss of function effect. So this shows the Sanger sequencing traces to confirm the mutation we identified. So it was a 4 base pair deletion in MBD4. This is our proband that had the large number of colorectal polyps. And we identified that both the parents were heterozygous for the same mutation. There's no known prior relationship between the mother and the father, no evidence of consanguinity. However, we do note that the parents were both from the same village in a fairly geographically isolated area. So whilst we can't rule out that, at some point further back in earlier generations, they may somehow be related. We also identified then the family there as a sibling of our proband who was also heterozygous for the same mutation. So in terms of phenotype, when we first became aware of this patient and they enrolled in the Corgi study, this patient had about 60 adenomas at age 36. They went on to develop further adenomas and ended up having a colectomy age 47. And then unfortunately, fairly soon after that, developed myeloid dysplastic syndrome, which quite quickly progressed to AML. So we had access to blood samples and tumor materials from this patient, and we confirmed with a TACMAN assay that the RNA expression was stable in blood and tissue samples. And we established a lymphoblastoid cell line to be able to look at protein levels. As you can see in the blot shown here, our patient is D21, and we were able to see there was complete loss of protein in the blood sample from the lymphoblastoid cell sample, compared to a cell line control where we knew there would be expression of MBD4. And there are problems with some of the MBD4 antibodies, so this also just shows we used an siRNA just to confirm the specificity of the antibody to MBD4. We also confirmed complete protein loss in the adenoma samples that we had available from the patient. So this top panel here showing loss in our patient compared to the normal colon control that was analysed alongside the sample from our patient. So we then went on to establish how common the mutation that we'd seen in our patient was. It's present in one Latino genome out of 250,000 alleles in version 2.1.1 of NOMAD. We checked whether or not the variant was at a high frequency in the area where our patient originated from, and in 100 population controls from that area, we did not detect any copies of that variant. And the variant was also absent from a series of 1,457 European polyposis and early onset colorectal cancer patients. So just to describe a little bit about what MBD4 does. So it's involved in base-excision repair. It's a DNA glycosylase, which is very important in the repair of DNA methylation damage. And in particular, in removing mismatched thymine and uracil when inserted or when present as a result of spontaneous deamination of the methyl C. And so normally MBD4 removes these and long and short patch repair can occur. And base-excision repair is known to be very important in colorectal polyposis and colorectal cancer already. Other genes involved in base-excision repair have been known to, mutations in other genes have been shown to cause other polyposis syndromes. So two recessive syndromes, MUT2H associated polyposis and MTHL1 associated tumor syndrome. These syndromes present with either polyposis, early onset colorectal cancer, and in the case of patients with the MTHL1 recessive mutations, extracolonic tumors as well. And MUT2H and MTHL1 are also associated with very specific somatic mutation signatures. So SBS36 for MUT2H mutation carriers and SBS30 for the MTHL1 associated tumor syndrome patients. And MBD4 has also been knocked out in mouse models and has been shown there to the mouse models confirmed this role that MBD4 plays in the repair of methylation induced damage. So as I mentioned, we've collaborated with a group in Melbourne led by Ian Majewski, and they first described biallelic mutations in MBD4 in 3 patients with AML. These were all diagnosed under the age of 35. And whilst the paper really characterises the changes that are seen in the AML tumours, which show very high mutation burden and a very specific tumour signature, which is mainly characterised by C to T mutations at CPGs, very similar to the aging signature known as SBS1 in the cosmic signatures. We also read with interest that these patients, at least 2 of the 3 patients they described, had also had a history of colorectal polyps, although at this stage that phenotype wasn't well known by the group. But all of these AML, these 3 AML carriers, all have biallelic mutations in MBD4, and DNMT3A mutations were seen in the tumor material, and also hotspot mutations in IDH1 and IDH2. And subsequently, we've worked with this group who've been able to obtain tumor material from one of their cases, which is known as WEHI2. We've now analysed 12 adenomas from this patient, and we've gone on to work with both this patient and the D2-1, as I'll describe, to look more at the tumor burden, tumor mutation burden, and also the signatures in the colorectal tumors to see whether we see something very distinctive, as was seen in the AML patients. So on the left-hand side here, to show 2 schematics of the colons of the 2 patients that we have access to. So D2-1 is the one that we've identified, and then WEHI2 is the one of the AML patients that was first described by the group in Melbourne. And we've obtained samples from multiple locations in the colon at several time points from each of these 2 patients. And have gone on to apply whole exome sequencing of the tumours in order to, as I mentioned, look at the mutation burden and spectrum. And as you can see from this panel on the right-hand side here, almost all the mutations are SNVs rather than indels, and compared to sporadic adenoma cases, which is shown here on the far right-hand side, they showed a much higher burden of SNVs. So on average, the D2-1 patient, where we had fresh frozen tumours from, as well as FFPE tumours, the number of mutations per megabase was on average about 11 per megabase, compared to only 1.8 in the sporadic cases. We saw that in the samples that we'd received that had been obtained at different time points, we could see increasing burden of mutations with age, as you might expect, in both D2-1 and in the WEHI2 samples. And as I mentioned, the vast majority of these mutations are C to T mutations at CPGs, as was seen in the AML samples first described by the Melbourne group. And when we compared the biallelic carriers, which is shown here in either the brown or the blue, and green, there was a much higher burden of the C to T at CPG mutations compared to both sporadic adenoma patients, shown in the pink, and also TCGA COAD samples as well. We performed methylation array analysis using the Illumina 450 K-array, and were able to overlay the sites of them that have been assessed for methylation with the sites of somatic mutation seen in the D2-1 polyps. And we were able to show that the majority of the mutations were occurring at sites of methylation. And this was not the case when we compared to some sporadic adenomas that we also had access to, analysed in the same way. So yeah, the signature that we see in both the polyps and the AML samples from the biallelic carriers is almost exclusively SBS1 signature mutations, and the de novo single base substitution mutation extraction from D2-1 only extracts this single signature. And as you can see here, the signatures seen in both the polyps from D2-1 and WEHI2 very similar to the signatures seen in the AMLs, and show very high cosine similarity to the cosmic signature SBS1 in cosmic signatures version 3. And yeah, over 90% of the mutations seen in all the colorectal samples from both D2-1 and WEHI2 were of this signature. And this next slide just demonstrates that as well. So compared to sporadic adenoma cases here on the left-hand side, signature 1 is in yellow here. You can see this much higher burden of SBS1 in our 2 biallelic MBD4 carriers. So we went on to have a look at driver mutations as well. So in the analysis of the AML samples that the Melbourne group had done previously, they showed very specific mutations driving the AML down a particular path. And there that involved DNMT3A and IDH1 and IDH2 mutations. So we wanted to investigate what types of driving mutations seem to be driving the tumors seen in these patients. And as you can see, APC mutations were very common in the biallelic patients compared to sporadic adenoma cases. And in particular, one particular codon, which is shown here, marked here by the pink boxes, was seen in 9 out of 10 polyps from D21 and all of the WEHI2 polyps. And this particular codon is the most commonly mutated in TCGA and TCGA coad and read samples shown in the panel below, but the frequency is much lower, so about 7% of patients have that hotspot mutation in APC, whereas nearly all of our samples showed this particular mutation. We did look at whether or not methylation levels were particularly high at this site, but that wasn't the case. There were other codons in APC as highly methylated, so we don't think that explains why this site was seen mutated in so many of the patients. We also see evidence of extra mutation. Oh, sorry, wrong direction there. We also see enrichment of missense mutations in Titan and also in AMA1 in the patients with the MBD4 loss of function mutations compared to the sporadic cases. And we see less KRAS mutations in the MBD4 patients compared to the sporadic cases here. And between 85 and 90% of the driver mutations from D21 and WEHI2 were these CPG and TPG mutations, whereas only about 37% of driver mutations in the 9 sporadic cases that we had access to were mutations of this type. So having identified that all of 3 of the 4 patients that with the biallelic MBD4 mutations had this rather unique phenotype of both colorectal polyposis and AML, we wanted to work out whether or not how often MBD4 mutations were causing this very rare phenotype. And through collaborations and through access to the UK Biobank data, we were able to identify 26 additional cases with either colorectal cancer, colorectal polyps, and AML or its precursor, myeloid dysplastic syndrome. And none of these 26 had either biallelic or monoallelic germline loss of function MBD4 mutations, suggesting that MBD4 loss is a rare cause of this very rare combination of phenotypes. So there have been a number of reports in the literature of monoallelic germline mutations in MBD4 being associated with a large increased risk of uveal melanoma. And this has been shown to be a strong enrichment of these MBD4 mutations in cohorts of uveal melanoma patients compared to population controls. But interestingly, what happens in these patients is that the tumours often have loss of the wild type MBD4 allele, and these tumours also display the same high tumour mutation burden and characteristic signature with the high number of C to Ts at CPGs that have been seen in the AML and the colorectal samples. So we wanted to have a look at whether or not those with single MBD4 mutations, whether we saw a loss of that second allele in the tumours of colorectal tumours as well. So we began this work by screening a large number of polyposis and early onset colorectal cancer patients for MBD4 mutations. This was done in collaboration with Richarda Dever's group and many others with cohorts of colorectal polyps and cancer cohorts. It also included screening polyposis cases that were sequenced as part of the rare disease arm of the 100,000 Genomes Project, and 283 cases from Genomics England were included in this cohort. And so in this cohort of 1,457 patients, we identified 5 cases with a single MBD4 loss of function mutation. We performed further long-range sequencing in these patients to see if there was a second pathogenic mutation in the germline, but we found no evidence of a second mutation in the germline. We had access to 4 tumours from these heterozygous carriers, and 3 of them showed no loss of the wild-type allele, and mutation burden was low to moderate, with 2 to 5 mutations per megabase seen in these tumours. And SBS1 did not fully explain the mutations apart from in 1 tumour. So I think we can conclude that the loss of the wild-type allele isn't happening commonly in the carriers that go on to develop colorectal tumours. And we also investigated whether or not the frequency with which we'd seen loss of function mutations in that cohort of colorectal cases and polyposis cases, whether the frequency in that cohort was different to the general population, for which we used NOMAD, non-Finnish European population as our comparison. And so we did see an enrichment in the polyposis cases, but we failed to see co-segregation analysis showing that the mutation was co-segregating with disease in these families. And also the mutation signature analysis is also not conclusive for a role of heterozygous MBD4 loss of function mutations in colorectal cancer polyposis susceptibility. Using the Genomics England data, we also investigated how common heterozygous loss of function mutations in MBD4 were. So we looked in the familial colorectal cases that have been collected by Genomics England, and also in the general colorectal cancer GSIP samples as well. And we identified 4 heterozygous loss of function mutations in the colorectal set. And this was in comparison to 21 heterozygous loss of function mutations that were identified in the rare diseases domain, where we specifically only included patients where there was no cancer-related phenotypes and only included unrelated individuals. And here we saw no association with risk for the heterozygotes. And there was no enrichment. We looked in UK Biobank as well, no enrichment for MBD4 loss of function mutations in patients with colorectal phenotypes or AML phenotypes. This next slide here also just shows that the monoallelic carriers, the heterozygotes, that's shown in blue here, have a much lower proportion of C to T mutations at CPGs compared to the biallelic carriers shown here in green. This is the D21 samples here. So we believe that biallelic germline loss of function mutations cause a rare recessive syndrome characterized by acute myeloid leukemia and adenomatous colorectal polyposis. It's a very rare syndrome with 1 in 5 to 6 million individuals having biallelic mutations in MBD4. The MBD4 deficiency results in somatic mutation spectrum that's very similar to cosmic SBS signature 1. And MBD4 we think should be implemented in gene panels used for the genetic testing of early onset AML or hereditary colorectal cancer and polyposis. And we propose 2-yearly colonoscopy from age 18 to 20 or the date of diagnosis, and regular follow-up with full blood counts in these cases, and genetic testing for any family member being considered as a stem cell donor. So just to summarize, the 4 biallelic germline MBD4 carriers discovered by ourselves and collaborators 3 of the 4 have developed colorectal polyps and AML. And these carriers with biallelic mutations are at high risk for colorectal polyposis and MDS AML. Monoallelic germline loss of MBD4 does not seem to greatly increase the risk of polyposis or colorectal cancer. And we do see very specific driver genes mutated in these patients with the biallelic MBD4 germline mutations. But as I highlighted, mutations in MBD4 and will only explain a very small proportion of those with colorectal polyposis or colorectal cancer and AML. But we think MBD4 should be added to gene panels used for genetic testing of early onset AML and also colorectal cancer and polyposis, where there's family history of these conditions. Yeah, thank you very much for your attention, and happy to take any questions.
Thank you so much, Claire. That was really interesting. Let's just see. So we have our first question from David Curtis in the chat. Are there any thoughts as to why defects in these mismatched repair genes tend to affect colon specifically rather than other dividing tissues?
Yeah, I mean, it is interesting why we're seeing this very specific phenotype in both colorectal and AML. And I think as often has been seen with other rare syndromes like this, it may be that as time goes on, we see that there is susceptibility to other tumours as well. It's really difficult when there's a rare number of patients with these syndromes. With these mutations. But one theory might be that APC, which is a large gene, is very susceptible to being mutated at the C to T, at cytosines that get spontaneously deaminated, and that means there's a higher chance of them going on to develop a colorectal polyp. But I think it remains to be seen whether these patients have susceptibility to other tumours as well.
Thank you. And we have another question from Jamal Nasir. So were you screening for just this gene, and is it worth screening all MMR genes? And I assume people are already doing this.
Yes, absolutely. So yeah, the mismatched repair genes are very commonly screened in all of the gene panels that get used for diagnostic testing. Also important to know for treatment choices as well. We know that those with mutations, both germline and somatic mutations in mismatched repair genes, respond better to different treatments. But we weren't just looking for mutations in MBD4 when we first screened that case. We had the whole genome sequencing data, but we did begin with a search on genes where we thought there might be there's some knowledge that those genes might be important in colorectal cancer. So we did look at all the genes in the base-excision repair pathway as well as those in the mismatched repair pathway. And this patient had already been through genetic testing at the centre that recruited it at St Mark's as well. So we knew that they'd already checked the mismatched repair genes and also APC as well.
Thanks, Claire. And I've got another question from AV Eid. So for the heterozygote risk assessment with the gel data, were you able to correct for age at inclusion? So when included at young age, or like no data on later tumour?
Yeah, so we did look to see whether there was a correlation between age and the number of C to T at CpG mutations. And yeah, there is a correlation with age, but the heterozygotes still showed a much lower burden of C to T mutations compared to the biallelic, and they didn't stand out compared to other tumours where MBD4, where there was no loss of MBD4.
Great, thank you so much, Claire. I think that's all our questions, but that was really interesting. And so if we move on to our next seminar now. Sorry, I think there might be one more question that's come through very quickly. I hope that's okay. So from Anupama Thacker, does the MBD4 loss of function mean that other proteins involved in the mismatch repair are not able to function too?
So it's known that MBD4 interacts with MLH1, and yeah, there may be some impact on mismatch repair that way. But we think the main reason that we're seeing a signature that's very specific to the MBD4 loss, it doesn't look like the signatures that we see don't look more like some of the mismatch repair signatures. But there will be some impairment of mismatch repair as well. But we think the primary problem is this failure to repair these sites of spontaneous deamination at the cytosine.
Thank you. Thanks very much, Claire. Great. So our next talk will be from Dr. Ian Wilson and Dr. Eric Olinger. So Dr. Ian Wilson works on the application of statistical methods to problems in genetics and has worked across population and ecological genetics, genetic anthropology, and the statistical genetics of rare and complex disease. His current work is on developing methods for assessing the impact of rare variants on complex disease. And Dr. Eric Olinger's work is primarily on tubular interstitial genetic kidney diseases and applications of genomic medicine in nephrology. His further work is on developing cellular models of kidney disease, as well as leveraging large databases for a better understanding of genetic nephropathies. And so this talk will be on the rare causes of kidney disease in the 100,000 Genomes Project. And similarly to our last talk, please save your questions for the Q&A session at the end. And we'll have some time for questions then. Thank you. Over to you, Dr. Wilson and Olinger.
So good afternoon, and thank you very much for the introduction. So my name is Eric Olinger. I'm a physician scientist working with Professor John Sire in the Newcastle University. And in contrast to Ian Wilson, as you can see, I don't have a bioinformatic background, but working mostly on clinical related questions. So thank you very much for giving us the opportunity to present some of the data or some of the things that we are doing in Newcastle. So let me see. So kidney disease is really a health priority with a prevalence globally that is around 10%, as you can see here on this map, and causes directly more than 1 million of deaths per year. And both the prevalence and the number of deaths are rising in the last years. In addition to this, it's also a major risk factor for cardiovascular disease. If we look at the etiologies of chronic kidney disease, then there are the major contributors like diabetes, but we also see that rarer causes like monogenic kidney disease or inherited kidney diseases contribute significantly to the burden of kidney disease, especially in certain population. And overall, we can estimate that around 10% of all adults with end-stage kidney disease and virtually all children ending up with end-stage kidney disease suffer from a monogenic or rare cause of kidney disease. Now, on the other extreme, the last years have shown that there's also strong genetic predisposition to chronic kidney disease in the general population, with over 300 loci being identified by genome-wide association studies. And what emerged also during the last years is a very interesting concept that there are shared pathways between rare diseases and loci associated with common diseases. So an example that is notorious in kidney disease is the humor locus, for instance. I show you here an example of something that is more familiar to what we are doing in Newcastle, which are the ciliopathies, which are rare, a group of rare disorders caused by a morphological or functional abnormality of the primary cilium, and which often come with kidney involvement. And this very recent study looked was applying a candidate gene approach to look at CKD defining traits in the UK Biobank, and was doing a particular lookup for these genes associated with rare diseases, ciliopathy diseases, and found that several of them were also associated with traits defining CKD in the general population. So showing this concept of shared pathway and amplifying why it is so important to study rare diseases, to understand and have more knowledge into pathways that are also applicable to common causes. And so to come back to the ciliopathies, which is our main focus here, the involvement of ciliopathy is called Nephronophthisis, which is an autosomal recessive disorder defined by renal concentrating defects, tubular interstitial nephritis, often corticomedullary cysts, and that progresses to end-stage kidney disease, usually before the age of 30 years, with different clinical forms, as you can see. The only firm diagnosis of nephronophthisis is a molecular one, However, it's not so easy because of the genetic heterogeneity. There are around 20 genes that are causing a pure form of nephronophthisis, and probably around 100 more of rare ciliopathy syndromes that in addition also give a kidney phenotype. Luckily, they are not all contributing to the same extent to nephronophthisis. So here's a study that was doing genetic analysis in a highly selected population of nephronophthisis patients and was able to molecular solve around 60% of them with ciliopathy genes. And among them, NPHP1 and NPHP3 were among the most common causes. And this is concordant with previous data showing that NPHP1 is responsible of around 80% of the pure nephronophthisis cases. Now, if we look in the Genomics England data set and we would select probants based on the status for chronic kidney disease and on nephronophthisis, and only very few with nephronophthisis, and would also select those where the father and mother are unaffected to enrich for an autosomal recessive inheritance, we find around 480 probants. But if we then check for those that are solved by either an exit questionnaire or tier 1 variants, under 15% of them, all these cases are solved. And among them that are solved for ciliary genes, seen here in blue, only very minority are solved for NPHP1 or NPHP3. So this shows, and these very preliminary results, that not only the diagnostic rate for these recessive or putative recessive diseases or these kind of disorders here is very low, but that also NPHP1 and NPHP3, which are major contributors to the phenotype, are probably underrepresented. And the reason for this might be the genetic mutations underlying these disorders. So for NPHP1, it has been known for a long time that due to repetitive flanking regions surrounding NPHP1, there's a strong predominance of large deletions involving NPHP1, and roughly 2/3 of all cases linked to this locus are caused by homozygous deletions. On the other hand, for NPHP3, we have been able to show previously that there's marked difference in splicing between the kidney and other tissues, and that rare synonymous variants that specifically affect kidney splicing might be the cause of kidney disease. So here we will see here that synonymous variants and large deletions are both classes of genetic modifications that present their challenges for next generation sequencing, as I will illustrate now by one short case, and then Ian Wilson will show you some other things that we have been doing for NPHP1. So this is a consanguineous family from Oman that is not in Genomics England, but it came to us via collaborators. And you see that the two siblings present with cystic kidney disease, with very early onset of end-stage kidney disease. And in addition, we're also suffering from congenital hepatic fibrosis. So altogether suggesting a ciliopathy. We have performed whole exome sequencing in the two siblings, but applying classical filters, there was no pathogenic variant that was identified or shared between them. Making use of the fact that this is a consanguineous family, we were then building a homozygosity map, where you can see in red and turquoise the two siblings and the regions that are homozygous in the genome for them. And on top of this map, so we were then appointing the candidate genes that have been known to be associated with this presentation, that is nephronophthisis and congenital hepatic fibrosis. And there are around 17 of them. And we can see here that NPHP3 is the only one that is mapping into a region of shared homozygosity. And then indeed, interestingly, when we went back to the whole exome sequencing data set and removed all the pathogenicity filters, we found a synonymous variant in NPHP3 that was shared between the two siblings that we confirmed by Sanger sequencing, including the segregation in the family. And this is a rare variant, only 2 copies in Nomad, and is predicted to have potentially an effect on splicing. So we were interested by this, knowing that NPHP3 is a subject to splicing altering variants. So we had access to RNA from whole blood from the sibling. And we were in fact able to show that this variant is leading to an activation of a cryptic splice donor site, interestingly in the middle of exon 20, and leading to an aberrant splicing, that you can see here. So we have amplified this region, and by sequencing the amplicon, could confirm that this splicing is occurring. And I show you here the gel electrophoresis, where you see the shorter transcript in the cases, but not in unaffected controls. Now, at this point, we noted that we would expect to see only the aberrant transcript, but we saw also a lot of normal transcript in patients. And while we hypothesized that this is due to these splicing differences between organs, we could not easily validate this hypothesis because we had no access to kidney or liver tissue. So at this point, our best guess to make our case more strong would be to find other candidates with the same variant. And here we turned then to the Genomics England database, and by querying in the raw data set exactly the same position that we identified in our proband, we found two further homozygous carriers of this same allele. And by looking into phenotype, so they have both been recruited into kidney domains, so cystic kidney disease or congenital abnormalities of the kidney and urinary tract. And interestingly, both presented with cystic kidney disease, early onset congenital hepatic fibrosis, so very much reminiscent of the first case, but in addition also developmental delay and a cardiopathy, so suggesting a more syndromic ciliopathy. We have in the meantime identified two more families in international collaboration with exactly the same variant and exactly matching phenotype, so that overall we think that we have solved 5 families with exactly the same synonymous variants, and are pretty convinced of the pathogenicity of these. Now, what is interesting is that despite the fact that both cases have been recruited in panel or in domains where NPHP3 is on the panel, this variant has not been listed in the tiering or the exomiser table, most likely because it's predicted a benign synonymous variant. And these cases have been labeled as unsolved. So I think this is one of the messages here, that it remains still different to apply correct variant prioritization. So I will stop here for the first short panel by summarizing that so nephronophthisis and related ciliopathies are the most important monogenic cause of end-stage kidney disease. These diseases and others, rare diseases, by being an extreme on the mechanistical and phenotypical spectrum, and important for us to study to understand better more common causes of CKD. So it is not always easy, as we see here, to get a correct genetic diagnosis, but it's still important to have it for patients and for research. And that one of the limitations that I was highlighting here is still the correct filtering and prioritization of variants. And I think there's still the case to do a deep phenotyping of the family, and there's still a place for manual curation of candidate genes. And in these cases, we were able, by this way, to find an interesting variant in NPHP3. But this was only possible with the help of the raw data set from Genomics England and the rich linked data and phenotypical data about the genome. So thank you for your attention. And at this point, I give over to Ian.
Thank you very much, Eric. Is that now we move over to Ian's part?
Yes, this is now my slideshow, so I can go.
Thank you.
Okay, so I'll just start with the same opening slide. I'm Ian Wilson, and as the introduction said, I'm a statistical geneticist. So what I'm going to talk about is how I've been trying to infer copy number variation in, well, it's in a few genes, but this particular talk I'm going to concentrate on NPHP1. So here's the Nomad browser coverage around NPHP1, and as we can see, they don't seem to, they look at the mean per base depth coverage of the whole genome for this is in the control individuals in the phase 2 copy number of the structural variation sample. So anyway, at various points of this, they are not getting any quality controlled coverage at all around NPHP1, which when I was thinking about looking for copy number variation was actually quite nice for me, because I've spent some harrowing times trying to infer copy number variation from SNP arrays about 7 or 8 years ago, and looking at exome sequencing and trying to find exome sequencing inferring copy number variants from exome sequencing, and to a large extent failing to find small copy number variants that are shared by both when you infer them from both exome sequencing and from SNP arrays, and in fact, failing to find them between failing to find them in families, and failing to find them in any sort of replicable way. So the general way of finding, nice bioinformatic way of finding copy number variants is to use a hidden Markov model and wander along the chromosome and share information upstream to information downstream, so that we can build a picture of where the copy numbers change. When we have areas where clearly alignment is not working very well, we can't, and things like that, then obviously that is unlikely to work well, because we're not getting any coverage. If we wanted to use breakpoints to find them, again, the alignment is not reliably putting things in the right place. So I thought we can do something very simple. We can just look at depth alone. So I chose a 1 megabase region around this particular around NPHP1, and this is just my example. As I said, I'm doing this on several different genes. So initial study: select all individuals in the renal domain with cystic kidney in the HPO term of the rare disease participant phenotype table. This gives me about 750 assemblies, of which 50 are repeated. Extract read depth of BAM files for a 1 megabase region around it. Gather read depth for all individuals. Thinned every 50 bases for in both of the builds, and look at the mean and variation. So a picture of this around NPHP1, here's about 10 individuals that I've got a heat map of the depth of coverage. So each one of these rows is an individual. There's the gene there. Each one of these rows is an individual, and we can see a very clear deletion of NPHP1 there. The heat map, 0, depth of 0 is white, a depth of about 100 is blue, and there's scale between them. So we can see that there's a deletion there. However, there's a lot of variation as we go along the genome. All these bars go white and dark. Here we see the regions that are not aligned particularly well, that have variable depth, spiky regions, both upstream and downstream of it, which is clearly the reason why we're getting deletions, that we have repeated sequences, but it also means it's very difficult it can be difficult to infer them. So that's my plot using GVIS. Now we'll look at all of them, all 494 in all the individuals in Human Genome Assembly 38. And while we do get higher depths from within GEL, within the 100,000 genomes in these regions that were not aligned at all, that didn't have good alignment in the nomad, they're very variable. I've also looked at the standard deviation of various other measures. This is just the simplest measure we've got. So what I chose to do is, if we go downstream about 400 kilobases, we have a nice region that is relatively stable. The means look about the same. The mean coverage looks about the same. Go upstream, the mean coverage looks about the same. So can we make inferences about the copy number within this central bit from the external ones in a very simple manner? So if we go back to the Nomad plotter, these are the two positions outside, and the green is the same over MPHP1. So after, as I talked about my difficult experiences doing exomes, inferring copy number variants from exome sequences, isn't it nice to have a well-sequenced human genome and the target mean depth? So the mean depth in the NPHP1 is plotted against the mean depth for each individual. The target mean depth over MPHP1 is plotted against the mean depth of 400 kilobases upstream and 400 kilobases downstream of a region of about 10 kilobases or 20 kilobases. So there's a lovely straight line. We can infer, if everything's working correctly, if the presumably if the copy number is correct, we can infer the target mean depth within NPHP1 by the mean of the flanking sequences over there, and we get a lovely straight line. However, we get a few individuals off the line. So what I now do is to standardize the target mean depth by the flanking sequences mean depth, so get the relative mean depth in the target region from the flanking regions. I should say that in the structural VCF files for NPHP1, none of these, I don't seem to be able to find any. And we see individuals with copy number 0, as in there is no depth in NPHP1. Here we see the relative depth as a half, implying there's one copy of NPHP1. And here we see ones that are very strictly around 1.5, so 1.5 copies of MPHP1. I haven't put any error bars on these. The calculations for error bars is going to be, since it's only calculated every 50 base, but there's still lots and lots of internal correlations and things. But additionally, we have a few that are just below 1, which, if things are if it is what I think it is, these are deletions of partial deletions of the gene, deletions of 1 or 2 exons from that gene. So I can see that in a plot. If I look at the mean depth across each one of these dots represents the mean depth within an exon on NPHP1, and we can see the red one are the 2 copy number 0s. Clearly the whole thing has been deleted, but we can see the inherent noise. The 2 green ones are, so this is the relative depth, the 2 green ones are 3 copies. So whereas they are clearly average 3 copies over this genome relative to the 2 flanking sequences, there's a lot of noise and difficulty to infer them. So I have had some success in doing this, which I'm not going to present here. You can see that there are things that look like deletions of individual exons around. So what do the copy number 0s look like? As we talked about, the first one is a nephronophthisis. Nephronophthisis, renal tract calcification, abnormal biochemistry looks like that. And 2 normalized specific disease, acute, many kidney phenotypic abnormalities which are shared between both of them. That's the phenotype that I see. And if anyone wants to ask about these in particular, I think I'll direct you to the clinician rather than me. So that's the end of my part of this talk. So acknowledgements in the Sayer Miles lab with John Sayer, Colin Miles, and Lisa and Sarah and Miguel and Laura. and lots of clinical collaborators. And as I said, this research is made possible through access to the data and findings generated by the 100,000 genomes, and the high quality of the genome-wide sequencing, enabling you to do enabling you to find these copy number variants relatively straightforwardly. However, when they haven't been found using other methods. Okay, thank you.
Thank you so much, Ian and Eric. So we have time for questions now. Let's have a look. Great, so the first question, does the 100,000 Genomes Project have phenotype data against every variant and other map details?
Yeah, so I think that the phenotypes are linked to the participant IDs. So at least the way I was using it for the NPHP3 variants is to identify the proband that carry these variants, and then to link the phenotype back to the proband, but not linking the phenotype to the variant. But I guess you can do some queries of phenotypes against particular variants. That's also possible to do these kinds of analysis.
Thank you, Eric. Just wondered if we have anyone at GEL as well that might be able to answer that question as well. In the meantime, we have another question from Catherine Bull. Why do you think the NPHP1 variant was not recognized as splicing in annotation? And how could we look for these kinds of misvariants more systematically?
I imagine that I don't know. My feeling about it is just that the quality control of the alignments and things upstream and downstream of it makes it very difficult to run the standard sort of copy number variant software to run over that. I mean, I didn't choose to try to write a copy number to use any of the standard copy number things. I didn't fancy trying to make it work through on the GEL environment, and I didn't really want to write it on the GEL environment, so I chose something very simple, which appears to work. But I think it had just to do with the repeated sequence, the duplications, the segmental repeats upstream and downstream make it very difficult to find breakpoints and to do it like that.
Thanks, Ian. Yeah, sorry, did I interrupt?
No.
And so another question from Obed Brew. Did you examine the mean depth in long or short reads? And if short reads, what measures did you take to rule the problem of inherent low map queues with short reads?
I haven't done that. No, I've taken the I've run a fairly strict QC. You know, I've used the general QC on them, and then just extracted the depths using SAM tools. I've done a very simple method on there. I haven't looked at any very long read data. I'm not particularly aware of where that is. I've just taken the standard BAM files and extracted sections of those, which are the standard read lengths. Yes.
Great, thank you. We have no more open questions. If there's any more questions, we have time. And If not, thank you so much to our speakers, Ian, Eric, and Claire, for your very enjoyable talks.
Thank you.
And just to let you know, we do have our next seminar, same time next month, so the 25th of May at 2 o'clock. And we're inviting talks from Dr. Yiting Zhang from Velo Health on patient subgroup discovery and insights generation through a multi-omics integration framework, and also from Dr. Matt Murray and Dr. Patrick Tarpey, who will be speaking about a rare case of pediatric astroblastoma with concomitant MN1 GTSE1 and EWSR1 PAX1 gene fusions altering management. And thank you again very much to our speakers, Dr. Pallis, Dr. Wilson, and Dr. Olinger,